Your cells are talking to each other right now, not metaphorically, not through chemical gradients or nerve signals, though those exist too, through tiny molecular packages so small that 100 of them lined up side by side would not equal the width of a human hair, that are constantly being released by every cell in your body and absorbed by neighboring cells and distant organs. These packages are called exosomes. And what they carry, what message they are broadcasting, is one of the most important determinants of how fast you age. Think of it this way. Every cell in your body is a broadcasting station, and exosomes are the signals it sends. In youth, those signals are predominantly regenerative. They carry microRNAs, tiny regulatory molecules, that tell receiving cells to repair their DNA, maintain their mitochondria, clear their cellular debris, sustain their stem cell populations, and resist the accumulating damage of daily biological wear and tear. The body, through its exosome communication network, is continuously telling itself to stay young, to repair, to regenerate. After 50, the broadcast changes. Aging cells, particularly senescent cells, damaged cells, and cells operating under chronic oxidative and inflammatory stress, begin producing a fundamentally different exosome population. Their exosomes carry different cargo, different microRNAs, different proteins, different signals. And the message those signals carry to receiving cells is not repair and regenerate. It is age, inflame, dysfunction. Research published in Nature Cell Biology has demonstrated that exosomes from aged cells, when transferred to young healthy cells, produce measurable acceleration of aging markers in the recipient cells. The young cells begin aging faster. Their mitochondria deteriorate. Their telomeres shorten more rapidly, their inflammatory profile increases, their stem cell activity declines. Your aging cells are not just failing themselves, they are broadcasting failure to every other cell that receives their signals. And the brain, with its extraordinarily active intercellular communication requirements and its dense population of exosome-responsive glial cells, is one of the organs most directly affected by this shift in the exosome broadcast. But here is the part of the story that changes everything. The exosome broadcast is not fixed. It is not determined solely by chronological age. It is determined by the biological state of the cells producing the exosomes, their oxidative burden, their inflammatory environment, their metabolic efficiency, their NAD+ levels, their senescent cell burden, all of which, across 25 videos, we have been working to optimize. And specific compounds, identified in research from leading aging biology labs, have been shown to shift the exosome broadcast from aging cells back toward a more regenerative profile, not by external cell therapy, not by injecting young blood or young exosomes, by changing the biological state of your own cells so that the exosomes they produce carry more regenerative and less pro-aging signals. By the end of this video, you will understand this mechanism completely, and you will have a practical protocol for shifting your cellular broadcast. Let's establish a precise understanding of exosome biology, because the details are important for understanding both the aging mechanism and the intervention. Exosomes are a subtype of extracellular vesicles, membrane-bound particles released by cells into the extracellular space and eventually into the circulation. They are formed inside cells through a specific process involving the endosomal pathway, a cellular trafficking system that manages sorting and recycling. When the endosomal system directs contents into multivesicular bodies, these inner vesicles can be released when the body fuses with the cell membrane. These released vesicles are exosomes. The size range is typically 30 to 150 nanometers, making them significantly smaller than any cell. Their small size allows them to circulate in the blood, cross tissue barriers, and critically, cross the blood-brain barrier to reach brain tissue. What makes exosomes remarkable as biological communicators is their cargo. Exosomes are not empty bubbles. They are packed with functional biological molecules. The most important cargo categories for understanding aging and brain health are microRNAs, which regulate gene expression in receiving cells. A single microRNA can regulate hundreds of target genes simultaneously, making exosomal transfer a powerful mechanism of intercellular gene regulation. They also carry proteins, including enzymes and signaling molecules, and mitochondrial components like mtDNA fragments that influence energy metabolism. The selectivity of exosome cargo loading is extraordinary. Cells do not randomly package contents. They selectively sort specific molecules for inclusion. The selectivity means that the exosome cargo reflects the cellular state that produced it, and changes in health produce changes in composition. When researchers analyze cargo from young versus old cells, they find dramatic differences in the microRNA populations. Young cell exosomes promote autophagy, mitochondrial biogenesis, and DNA repair. Old cell exosomes promote inflammation and cellular dysfunction. The exosome microRNA landscape is essentially an epigenetic aging signal, a broadcast of the cellular state that propagates aging phenotypes. Now let's trace the specific pathways through which aging exosome signals spread cognitive decline after 50. The first pathway involves exosomal transfer of pro-inflammatory microRNAs from peripheral aging cells to brain microglia. Research established that exosomes from peripheral aging cells, particularly aged adipose tissue, carry specific microRNA cargo. When absorbed by brain microglia, this cargo shifts the phenotype toward the pro-inflammatory SASP secreting state characteristic of neuroinflammation. The microRNAs transferred, like MIR146A and MIR155, directly regulate the NF-κB inflammatory signaling pathway in microglia. This mechanism propagates neuroinflammation into brain tissue through the exosomal communication channel that crosses the blood-brain barrier normally. The second pathway involves exosomal suppression of neural stem cell activity in the hippocampal subgranular zone. Research found that exosomes from aged blood significantly suppressed hippocampal neural stem cell activity in adult neurogenesis. This finding reveals an exosomal mechanism for the age-related decline in memory formation and cognitive flexibility. The third pathway involves exosomal transfer of damaged mitochondrial components, such as dysfunctional DNA fragments. When neighboring cells absorb these exosomes, they experience a transfer of mitochondrial dysfunction. Their energy production falls. In the brain, where energy demands are extreme, this transfer is a significant accelerant of cognitive decline. The fourth pathway is exosomal modulation of BDNF signaling, specifically reducing a TRKB receptor expression in receiving cells. The aging exosome signal creates a BDNF resistance in neurons, where increasing BDNF production is offset by fewer receptors. Optimizing the exosome environment amplifies the BDNF protocol's effectiveness by ensuring the brain remains responsive. Let's go deeper into brain exosome biology. Neurons in glial cells have specialized mechanisms that create unique vulnerabilities and unique opportunities. Neurons are among the most prolific exosome producers. Their complex morphology, with axons extending long distances, requires constant intercellular communication for maintenance. Exosomes are the primary vehicle for this long-distance communication. Neurons release them from the cell body and axonal processes to signal surrounding glia. The cargo of neuronal exosomes includes synaptic proteins, neurotrophic factors, and microRNAs that regulate gene expression in recipient cells. In healthy young neurons, this cargo promotes synaptic maintenance, supports myelination, and maintains the neuroinflammatory balance for neuroprotective functions. As neurons age, particularly under oxidative stress and metabolic impairment, the cargo of neuronal exosomes shifts significantly. Stressed neurons produce exosomes enriched in pro-apoptotic signals and depleted of neuroprotective cargo. Recipient neurons then show accelerated aging markers. The tau connection is critical. Pathological, hyperphosphorylated tau, which forms Alzheimer's tangles, spreads through the brain via exosomal transfer. Neighboring neurons that absorb these tau-laden exosomes develop tau pathology themselves, creating a primary mechanism for the progressive spread of Alzheimer's. When brain insulin resistance is high, neurons produce more tau. When autophagy is impaired, the tau is not cleared and spreads to neighbors. Oligodendrocytes, the glial cells maintaining myelin sheaths, are also vulnerable. Their exosome production is essential for neural signal insulation. Aging oligodendrocyte exosomes carry increased stress signals that impair myelination. This contributes to the myelin deterioration seen in aging white matter. This deterioration impairs neural signal speed, contributing to the processing speed decline that is a consistent cognitive change of aging. Now let's talk about the compounds that research supports for shifting the exosome broadcast from aging toward regenerative. This is where the science moves from understanding the problem to providing the solution. The approach to exosome optimization through natural compounds is relatively new, and the evidence base, while compelling, is less mature than for many interventions. What follows represents the best current evidence, but it is evidence that will be refined by ongoing research. Compound 1, urolithin ELA. You already have urolithin A in your protocol for its mitophagy-activating effects on damaged mitochondria. The exosome biology provides an additional and powerful rationale for its inclusion. Research in Nature Metabolism found that urolithin A treatment in aged cells produced significant improvements in the cargo of exosomes released by those cells, specifically increasing pro-mitophagy and anti-inflammatory microRNAs in the exosome payload, while reducing the pro-inflammatory cargo characteristic of aged cell exosomes. The treated cells, exosomes, were broadcasting a more regenerative signal, which promoted mitochondrial quality control and reduced inflammatory activation in neighboring cells. By improving mitochondrial quality in the producing cell, urolithin A reduces the cellular stress state that drives the shift toward pro-aging exosome cargo. Healthier cells produce regenerative exosomes. Urolithin A makes cells healthier. Therefore, urolithin A improves exosome cargo through the mitochondrial quality pathway. Additionally, research found that urolithin A promoted the production of exosomes enriched in SIRT-T1 protein, the NAD+ dependent longevity enzyme. Exosomes carrying SIRT1 deliver this anti-aging enzyme to recipient cells, extending the benefits of urolithin A's cellular effects to neighboring cells. Compound 2, quercetin at senolytic pulse doses. We've established quercetin for anti-inflammatory and senolytic effects earlier in this series. Research in aging cell found that quercetin treatment produced dramatic improvements in exosome cargo quality through the senolytic mechanism. By eliminating senescent cells, the primary producers of pro-aging exosomes, quercetin removes the most toxic exosome signals from the tissue environment. Senescent cells release exosomes enriched with SASP signals, broadcasting dysfunction. When quercetin eliminates them, the signal shifts toward regenerative. This exosomal mechanism explains why senolytic therapy produces such broad anti-aging effects across multiple organ systems. Compound 3, spermidine. You have spermidine in your protocol for its autophagy activating effects. The exosome science adds an important additional rationale. Research in Nature Communications found that spermidine significantly improved the regenerative cargo of exosomes released by treated cells. Spermidine treatment increased exosomal microRNAs that promote BDNF receptor expression and suppress inflammatory NF-kappa B signaling. Research from Graz found that spermidine supplementation improved blood exosome cargo quality at the whole organism level. The exosome optimization additions are elegant, because all 3 compounds are already in your protocol from earlier videos. Understanding this mechanism allows you to appreciate why the synergies between autophagy, senolytic therapy, and mitophagy are so strong. However, one additional compound is worth adding specifically for its exosome optimizing effects: phosphatidylserine. Phosphatidylserine is a phospholipid that supports the healthy endosomal sorting function that ensures beneficial cargo is loaded into exosomes. Additionally, phosphatidylserine has direct cognitive benefits through its role in neuronal membrane integrity and neurotransmitter release. Multiple clinical trials demonstrate improvements in memory and cognitive performance in adults over 50. The research supported doses 300 milligrams daily, taken with fat-containing meals. Take with breakfast alongside your other fat-soluble supplements to optimize your exosome broadcast starting today. 26 videos. And with exosome biology, we have revealed something that fundamentally changes how you understand the entire system we've built. Every intervention in this series, every supplement and behavioral practice, has been described in terms of its direct biological effect. Lion's mane supports BDNF. Magnesium L-threonate rebuilds synaptic density. CoQ10 optimizes mitochondria. Sleep activates lymphatic clearance. All of that is true, but the exosome research reveals a deeper mechanism operating beneath all of these direct effects. When you improve the biological state of your cells, you are not only improving those cells' direct function, you are improving what those cells broadcast to every neighboring cell. You are improving the exosomal communication environment of your entire body. Better mitochondrial function. Through CoQ10 means healthier cells, more regenerative exosomes, and neighbors that age more slowly. Better autophagy through spermidine means cleaner cells and better exosome cargo. Lower inflammation through omega-3 means less stressed cells. Every intervention produces a ripple effect through the exosomal network. The total biological impact is greater than the direct cellular effects alone. This is the exosome multiplier. It operates silently beneath every other mechanism in the series, amplifying the effectiveness of the entire system. The senolytic protocol from video 21 produces the most immediate improvement in the tissue exosome environment by removing pro-aging broadcast stations. The NAD+ protocol improves the quality control of exosome cargo sorting. SIRT-T1 activated cells produce more selectively regenerative exosomes. Circadian clock restoration ensures exosome release follows its natural rhythm. Clock restoration restores the temporal precision of the broadcast. Healthier gut immune environment means healthier immune cell exosomes, leading to less neuroinflammatory signal reaching the brain. 26 mechanisms, all connected through the exosomal communication network that permeates every tissue and organ in your body. Your action plan: add phosphatidylserine at 300 milligrams to your morning protocol. Ensure urolithin A, quercetin, and spermidine are consistently included. Take a moment to appreciate this. You have built a protocol that operates at the level of DNA, molecular machinery, and whole body biology. This is the most comprehensive approach to cognitive aging that science supports, and you are implementing it. Next week on Neurovitality. After 50, we investigate the impact of electromagnetic field exposure on brain health. We'll separate science from noise. Subscribe now, turn on notifications, stay sharp, Stay regenerative, stay vital. I will see you next week.